So in my last section, I'm going to talk about the cell cycle and cancer. I'm going to kind of bring everything together that we've talked about so far. The gene regulation, protein regulation, what happens with mutations, and I'm just going to bring it all together now into this final little package. So remember in the last section, we were talking about what happens if there's mutations that evolve in germline cells and how this can lead to basically evolution, huge changes throughout their generations to come. But now we're going to look at the other half of things. What happens when there's a mutation in a somatic cell that starts dividing and passes this mutation on to offspring within a single person? All right, so it's not being passed on generation to generation, but is being passed on through generations of somatic cells. So before we get into talking about cancer and these mutations specifically, we have to talk about the cell cycle and what controls the cell cycle, those specific mechanisms and physiology that controls whether or not your cells divide or don't divide. It's very important that your cells really control this strictly in order to prevent cancer. Remember we said that humans have 23 sets of chromosomes for a total of 46. And remember, every single cell in your body that has a nucleus has every single one of these chromosomes. And it's very important that all of your cells maintain these exact number of chromosomes and the exact genetic information. Otherwise, things like cancer could happen if there's a mutation in one of these chromosomes. Your body uses two different processes in order to divide your cells. The first process is called meiosis, and meiosis is when you need to make copies of sperm or eggs, otherwise known as gametes, the reproductive cells. We're not going to talk about meiosis today. Today, we're going to talk about mitosis. So mitosis is production or replication of all somatic cells. So somatic cells are all the cells in your body that are not the gametes. So anything that's not a sperm or an egg. This includes skin cells, red blood cells, white blood cells, bone cells, all right, everything else. Now, how mitosis works is you start out with the original cell. This is called the parent cell. And the parent cell has the nucleus here that contains all of those chromosomes. Then what happens is when the parent cell needs to divide, the nucleus is going to replicate itself. So you need to have now two nuclei that are both identical to one another. And this is very important so that the new cell is identical to the parent. After that, the cytoplasm is going to divide in a process called cytokinesis. And then we're going to end up with two daughter cells that were identical to the original parent. So each of the two new daughter cells are going to have exactly the same chromosomes and the same number as the original cell. And this is what makes mitosis different from meiosis. In meiosis, you have variation, genetic variation, which is why children don't look identical to their parents because the sperm and eggs have variations from each other. Whereas in mitosis, all the cells need to be identical. So for example, your skin cells are constantly dividing because your skin is shedding at all times. You need to make more copies of it. Or if you had an injury, like if you had a cut in your skin and your cells need to replace the worn out or dead ones. Also, your red blood cells are constantly recycling and new red blood cells are being formed. And your white blood cells and your muscles if you're growing muscle cells, things like that. Mitosis relies on something called the cell cycle. And the cell cycle has three main stages. The first stage is called interphase, and this is the longest stage in the cell cycle. Now, interphase is divided into three more stages. It's divided into G1, S, and G2. So the gray in this circle is interphase. The second stage is actually the mitosis stage, which is shown in this little colorful piece of the pie. And then finally, the third stage is cytokinesis, which, like I showed you before, is when the cytoplasm actually split. Let's talk first real quick about interphase. So interphase starts with the G1 or growth 1 phase. During G1, now you have a cell. You can envision maybe a skin cell or a red blood cell. First, all of the organelles need to double in number. So like the mitochondria, the ribosomes, lysosomes, things like that all need to double. And then the cell needs to accumulate material needed to make DNA. And finally, the cell needs to decide Should it divide or should it not? If it's a skin cell or a red blood cell, probably yes. If it's a neuron, neurons can't divide. You're born with all the nerve cells you'll ever have. So neurons don't pass the G1 phase. After the G1 phase, now the cells go into S phase. This is also part of interphase. S stands for synthesis because this is the stage where DNA gets synthesized. So, for example, in your cell, you originally had 46 of these chromosomes, but during this stage, it's going to double to 92 chromosomes. Finally, G2 phase, this is the last stage of interphase. This is when the cell decides, yes, we should definitely divide, and it's going to produce any proteins or enzymes that are needed for cell division. Now the cell's finished interphase, it's going to enter into mitosis. Mitosis has basically 5 steps, all right? So this image is showing you interphase, we had 46 chromosomes. After the S phase, we now have 92. So during prophase, you're going to start to see the chromosomes condensing, and all of a sudden the nucleus is going to start breaking down a little bit. What's going to happen is this thing called a spindle, it almost looks like a spindle from a spider web, is going to form. So by the time you get to the second stage, prometaphase, the chromosomes are now starting to line up in the center, but the nucleus has disappeared, the nucleus has dissolved, and the spindle is fully formed. And what this spindle is going to do, it's going to kind of, I like to think, grab these chromosomes, and it's going to line them all up into the center of the cell. And when they start lining up, this is when you're officially in metaphase. Now in anaphase, the spindle, those spider web things like sticky spider web, is going to pull the chromosomes apart. So half the chromosomes are going to go to one side, the other half to the other. But remember, each half is getting an identical set of chromosomes. Finally, we enter telophase, the final step. Now the spindles are going to disappear. You're going to see each half of the cell is going to have an identical copy of chromosomes. Now the cell is going to start to kind of stretch apart here in the middle, and eventually it's going to pull apart during cytokinesis. So that's the steps of mitosis. It spells PMAT. Prophase, prometaphase, metaphase, anaphase, telophase.